Thank you so much, Licia. And last up for our morning session, we have Sofia talking about the reef fish assemblage at a marine national park.
Hello everyone, my name is Sofia Ferreira. Today I'll be talking about a small portion of my master thesis looking at reef fish community assemblage. So I'll be talking a lot about structural complexity, and what I mean by that is the physical three-dimensional arrangement of non-living components, including rocks, boulders, and underlying topography, as well as living components, including corals, because they secrete calcium carbonate skeleton, thus providing a lot of intricate structure to the reef. Why we care about structural complexity: it provides a lot of coastal protection and boosts tourism revenue. But most importantly, it increases biodiversity by providing shelter, breeding sites, foraging grounds, and refuge for thousands of marine species. Unfortunately, we are rapidly losing this three-dimensional structure due to steep declines and community shifts in the coral assemblages, which is then compounded by ocean acidification, causing a rapid erosion on the remaining calcium carbonate structures. As you would expect, this is then resulting in declines in biodiversity, ultimately resulting in a decline on ecological functions and ecosystem services. This is now beginning a shift in focus on coral reef ecology from preserving particular species to make sure we can maintain ecological functions and services. And so this points us to fish, because fish play a critical role in maintaining key functions, including herbivory, bioerosion, predation, and nutrient release. So to effectively conserve transition in reefs, we need to understand reef fish community assembly in a manner that correlates to their crucial ecological roles. So trait-based ecology provides an avenue to do that, because functional traits are proxies to ecological functions. So literature shows that reef fish can exhibit a variety of ecological, morphological, and behavioral traits that can be linked to critical roles that they fill in the reef ecosystem. So while there are many studies looking at reef fish species relationship with habitat structure, I wanted to investigate this relationship by focusing on these traits. Because if we do so, we can better understand the role they play in the community, and then we can better understand how we can conserve functional reefs in the future. So then my main research objective was to evaluate the influence of photogrammetry-derived structural complexity and coral morphology on the trophic, behavioral, and morphological traits of reef fish. My study site was Kaloko-Honokohau Marine National Park in Kailua Kona. The dashed line represents the park boundaries, within where I had 19 sites. At each site, I had a plot of 125 meters squares. I began my survey by first laying my 25-meter transect and conducting a 10-minute bell transect survey, recording every single species and counting how many individuals per species I saw. Using the literature, I then assigned trait values to each of the species. Again, those traits were max body length, trophic group, crypsis, mobility, body shape, water column position, gregariousness, and activity period. After my fish survey, I conducted a photogrammetry survey. This involved swimming on top of that reef substrate in a back and forth motion, taking continuous and overlapping images, which allowed me to build high-resolution three-dimensional reconstructions of each of the reef sites. I then imported these models into the geospatial software, GIS, which allowed me to annotate the coral community. This is an example of one of the 19 plots. Every single polygon you see here represents an individual coral colony that I manually delineated. For each coral colony, I assigned a morphology, and I also sized the corals by the total three-dimensional area. Throughout the 19 sites, I annotated over 83,000 corals. For 3D structural complexity, I used the digital elevation model, which is another output of the 3D modeling process, which allowed me to compute the following metrics. Curvature measures the concavity and convexity of the surface. Slope is a change in elevation. Surface complexity is the ratio of 2D over 3D surface area. And vector terrain ruggedness uses vector dispersion analysis to compute how complex or flat the terrain itself is. Okay, so going into the results, here we're looking at the fish community-wide functional space. This is a PCOA, principal coordinate analysis, and it's providing a visual representation of how the species disperse in the functional space. So every single point here represents a species, and where they fall on the graph is telling us how similar or dissimilar species are based on their functional traits. So the points are colored by functional groups, which I assigned using hierarchical clustering analysis. As you can see, I resulted with 11 functional groups. And what I noticed was that the traits of gregariousness, water column position, and body shape were the key traits driving these functional groupings. As I mentioned, this is the community functional space, which represents the highest functional potential a given site could have. So if you look at some of those sites, some of them look like this. So in the blue here, it's one of the sites. You can see it fills up a lot of that functional space. It has high functional richness. Other sites look more like this. You can see it fills a lot less of that functional space. It's certainly missing a lot of those functional groups, which leads me to my question, can habitat structure help us understand these differences in functional communities? The short answer is yes. The abundance of functional groups are influenced by both 3D habitat structure and coral morphology. I will go through each of these models here in a little bit, but basically we're looking at the results of various generalized linear models. And on the left figure, we have the models that had the predictor variables being 3D complexity. And on the right figure, the predictor variables were coral morphology. The y-axis is showing us incidence rate ratios, and they show the effect of that predictor on the abundance of a given functional group. So if the value of the incidence rate ratio was greater than 1, that predictor had a positive effect on that group. If it was less than 1, it has a negative effect. Okay, so starting with one of those groups, functional group number 7 had schooling species with a short or deep body form, which showed positive responses to branching corals and cresting corals and slope values. On this group, we had species like the Hawaiian damselfish and also chromis, which is another damselfish. These species are very, very commonly found within branching corals. So that definitely explains here the branching relationship and possibly the slope as well. We did also have some herbivores on these species, which often like more encrusting areas where they can better access that algae. Functional group number 11 had nocturnal and cryptic species, showing positive responses to knobby, branching, and crusting corals, as well as higher slope values. The slope relationship here is no surprise if you've ever seen these species on the reef. It's pretty much always hiding underneath ledges and overhangs, which are areas providing that slope. These areas with those type of overhangs are also areas often dominated by encrusting corals, which we can also see that relationship here. Functional group number 10 had elongated cryptic species like the eel and the cornet fish. We see that it responded positively to surface complexity and negatively with curvature. I do want to emphasize here that that doesn't necessarily mean a negative relationship, because negative values of curvature mean concavity, positive mean convexity. If you've ever seen these eels on the reef, they like to hide inside of holes and crevices, which are areas that are providing a lot of that concavity. Cornet fish here, it's while elongated and cryptic, it does have a very different way of exhibiting that cryptic behavior. Instead of hiding within the reef, it is usually higher up in the water column, kind of blending in with the water. So that sort of explained why we had such a wider confidence interval with the corals here. Functional group number 1 had solitary mobile species swimming lower in the water column. We had a lot of herbivores and invertivores on this group, and we see that they had a negative response to VRM and knobby. The literature shows that herbivores usually respond in a negative way to very small scale complexity because again, they're herbivores, they're trying to look for broader, more open areas where they can easily access the algae. In a similar way, functional group number 3 had solitary fish with a fusiform body shape. Here we had more larger piscivores like the omilu or the jobfish and also larger invertebrates. In a similar way, we see that they had a negative relationship with VRM and branching. Again, these things provide very small scale complexity, and because these fish are larger piscivores, they can't really get in there to access their prey in that such like complex areas. However, they did have a positive relationship with slope, which could explain that these areas may be more favorable for them to go access their prey. So I don't have time to go through every group, unfortunately. But the key findings here are that in terms of the metrics, slope, surface complexity, as well as morphologies of branching and mounding, did seem to be the most influential structural features in the functional groups. And what I mean by that is that these metrics show the most numerous and the most significant relationships with several of the functional groups. Again, I couldn't go through every group, but 10 out of the 11 functional groups did show significant relationships with either complexity or coral. In terms of the fish, behavioral and morphological traits exerted a strong influence in defining the fish functional space and groupings, which is very interesting because the literature tends to neglect these traits and focus primarily on trophy groups. But as I can see on my study, behavioral and morphological traits exerted even a stronger influence in defining the functional structure than trophy groups did. So the broader impacts of the study, as I mentioned at the beginning, we're rapidly losing three-dimensional structure. And so these findings can help us understand not only what species we will lose, but what functional groups and how that may relate to the overall ecosystem functioning. Guiding restoration efforts, as we know, coral restoration is huge right now. So this can help us guide sort of what structural features we should restore, not only to maintain, to restore abundant coral communities, but ultimately, how do we restore fully functionally diverse coral reef ecosystems? Traits, interplay, and functional structure, as I mentioned before, most studies primarily only include trophic groups. So by me including all these different types of traits, I could see like, okay, what are sort of the traits that, how they interact together, and then what ends up driving that functional structure. And then lastly, metric standardization. With photogrammetry and all these new technologies, everyone is coming out with new metrics, which is good, right? Because all these technologies allowing us to do so. But it also makes it really hard to make generalizations across studies. So by including so many metrics of complexity, various morphologies, and various traits, these findings provide insights as to what may be the most influential things and what are things that we should be focusing on when we're monitoring the reefs and when we are restoring reef ecosystems. So with that, I'd like to thank everyone here, my advisors and NPS, and hopefully I have time for questions. Thank you. Yeah.
So Hawaii is known for sort of disharmonic biota, missing certain functional groups, especially in terrestrial systems. Are you seeing that in the fish where there's some trophic or some functional groups that are not here in Hawaii that are known elsewhere?
That's a good question. I don't really have an answer for it because there's not really other studies looking at this yet, especially with these many traits and stuff. So like the functional groups is just different because they included different traits, and then they didn't measure complexity like I did. So that's why I'm saying like it's kind of hard to make generalizations at this point because everyone's like starting to do kind of different things so that we can find out better. Yeah.
So great talk. I have two questions for you. first one, I guess, how do you separate out habitat from the effects of habitat on being able to detect these fish? Because as you increase the structural complexity of the habitat, you're going to be influencing your ability to reliably see and count these fish at the same time. Especially individuals and species that are very cryptic, like your eels or your polycentrids that are only active at night. So how are you, I guess, separating out that? Because it seems like that would be really tightly correlated.
Yeah, that's also a really good question, and I also don't have an answer for that, because it is true, and there's a lot of literature to show that we are drastically underestimated the abundance of these cryptic and nocturnal species. And so that's hard, and like I said, there's only one functional group that was not predicted by complexity, and it was actually the sedentary cryptic species, which doesn't make sense in a way, because you would think that those would be the highest, right? But because we undercount them so much, we're not even fully able, I think, yet to detect truly like how much there are of them. So I think, like, honestly, the best way to do that would be with environmental DNA, because then we can get a better understanding of how much of them are there, and then correlate that to habitat. But yeah, I think I definitely missed a lot of that in my study, unfortunately, because it's hard to see them.
And then I guess my second question is kind of related to that, is how did you account for scale as well? Some of these species, your survey area would represent the entirety of their world, and other species, it's a very small fraction of their daily core use area that you're looking at. And so I guess, how do you account for that in this sort of analysis?
Yeah, the only way I sort of accounted for that was using the mobility trait. So just kind of digging through the literature to see how mobile and what is the home range of these species. But then, of course, that would mean that, like I had 3 values in that category. I had sedentary, mobile, and wide. And I had very few, like maybe 2 species of wide, because again, like they're not really there as much. They're swimming a lot, so we're not seeing them too much. So then, yeah, that's sort of another thing that I think environmental DNA would be better. But yeah, I hope that sort of answers.
Thank you.
Any more questions? Well, thank you.
Thank you all for being here this morning. That concludes the first half of our symposium. We are going to break for a short lunch. We'll be meeting back at 12:45. So in the meantime, have some food. Check out the silent auction over here. Oh, and posters are just down the hall across the way through the glass doors. There's some posters that went up yesterday, I believe. Feel free to check them out. They're super cool. Thanks.
Thank you.
Thank you.